Okay. So greetings, everyone, and welcome to EMN Webinar series. My name is Thomas Dussarrat, and together with my colleague Diana Pinto, we are really pleased to, and the rest of the EMN, we are really pleased and to welcome you today, and we are happy that you could participate despite this so short time inconvenience. March is always a tricky month for time shift across the board. But yeah, so today the webinar will be about Untargeted Metabolomics for natural products, not only for discovery, but also for functional analysis. And before I will have the pleasure to introduce the speaker, I would like to encourage you to follow the EMN and MedSoc accounts on Blue Sky, X, LinkedIn, or simply via email. And if not, why not trying to motivate you to join our team? We are doing plenty of meaningful activities like this webinar, but also some work for the conference and many different activities for the Metabolomics Society. So do not hesitate to contact us. So today, we are really pleased to welcome Prof. Dr. Kang from Sookmyung Women's University. So his lab is trying to answer various questions on specialized metabolites, like natural products, for instance, using cutting-edge computational and mass spectrometry tools. He received his bachelor's in pharmacy from Seoul National University in 2010. And he then earned his PhD degree in natural product chemistry from Seoul National University under the supervision of Prof. Sookmyung in 2016. After postdoctoral work at UC San Diego, under the supervision of Peter Dorenstein, he founded his own lab in 2018. So he will now present this webinar about, as I said, untargeted metabolomics for natural products, not only for discovery, but also for functional analysis. And we will then pass the show to his PhD candidate, Pham, who will continue the presentation. So do not hesitate to ask questions in the chat, all the questions, or at least we will try to tackle all the questions at the end of this webinar. So thanks again, Prof. Kang, for accepting this invitation. And now the floor is yours.
Okay, thank you for introducing me so kindly. And then I'm gonna share my screen now.
Yeah.
Right, so my screen is shared, right? All right, so today I'm gonna talk about my recent works. So yeah, title is as shown in the screen, like Untargeted for Metabolomics for natural products. So, and I also added up a specific praise in here, like not only for discovery, but also for functional analysis, because I wanted to highlight like how we're actually using the metabolomics in our lab, or especially for, finding out some like a function of like enzymes or the proteins related to the natural products in a very, the various ways. So my lab is working on the natural product, and then it's also known as a specialized metabolites or as secondary metabolites. Well, I think most of you, of course, know what the natural products are, but we also call it traditionally like a secondary metabolite because in old days, scientists that thought like, well, natural products does not play an essential role in the biological organism for sustaining their life. So they thought like, okay, it's not very like a essential metabolite. But now we know that actually the organisms makes up the natural product for their specific like a reason to fight against their like a stress. For example, like the biological stress from like predators, or abiotic stress, for example, like the temperature, or like a drought stress, and or high salinity, and the others. So mainly they have actually their own drawer, and then we also call it like a specialized, because biosynthesize for such types of metabolite are actually really well specialized in each organism. Means that you can find out the metabolite A in the organism A, but you cannot find it out in the other organism. For example, you know that you can find out caffeine in the coffee or tea, but you cannot find them in the different types of plants, because their biosynthetic pathway is quite well specialized in the really specific organism in the phylogenetic tree. Anyway, so such kind of like specialized pathway makes the structures of natural products so complex. And then, so if you want to look into them through the untargeted metabolomics approach, the chemical complexity of natural products make them a really challenging subject of the untargeted metabolomics, because you're gonna see a tons of different mass spec signal in your data set, but it's really hard to annotate these structures, because, you're not gonna, if you're working in the centralized metabolism, for example, like the glycolysis, or like energy metabolism in the human, you can actually expect like what types of metabolites we're gonna in your data. But if you're working in the natural product, it's more challenging, because you're gonna have no idea about the expected structure of a metabolite in your data set. So mainly the metabolomics, the pipelines in my lab is quite well about the qualitative rather than in a quantitative way. And anyway, so yeah. But still, alternative metabolomics is quite well valuable the approach for a natural product chemist, because it will gonna give you tons of information about the chemicals in your sample, even without isolating them through the chromatography. So there are a bunch of papers describing how the untargeted metabolomics can be really like a valuable solution in the natural product discovery. But today I'll not talk about the usage of the metabolomics for a natural product discovery itself, but I'm going to talk about a little bit different application of such a methodology in the natural product researches. And actually, my talk in here, we're going to talk deal with about the biotransformation of a natural product, and the after my talk, the Huong, a PhD student from my lab, we're going to talk about a little bit different theme about the like ecological function, unless it's using the metabolomics. And here I'm going to talk about the xenobiotic biotransformation. And normally it means that the metabolism of an organism dealing with a chemical which is not made by the, the how do I say, like not made by the organism itself. For example, if you take some food or a drug, they're gonna have some chemicals which is not present in the human body in the naturally. For example, like most of the chemical drugs or in the food, there's some kind of like the plant natural products were gonna be there, or even some types of like a pesticides that are going to be there. They're going to just absorb it into our body, and then they're going to be a metabolism against such kind of chemicals. For example, your liver, we're going to metabolize the compounds into different types of compounds. All of you are going to know that if you just drink the alcohol, and then the ethanol, we're going to be absorbed here, and the liver, we're going to metabolize them into the acetaldehyde and acetate. It's another type of forms of the xenobiotic biotransformation. And it's also already really well known that our gut microbiota is another really another important driver of xenobiotic biotransformation. For example, so a lot of cases are already known. For example, like they're gonna just change the structures of the drug inside of the gut, and then it's gonna change the efficacy or the toxicity of the drug it takes. So currently, my lab is working on the Pham, and we're using the mass spectrometry based on targeted metabolomics for finding out the phenotypes of xenobiotic biotransformation happening in our body or driven by different types of microorganisms, so on. And so yeah, I'm gonna talk about this detailed case a little bit later. And I need to highlight that the xenobiotic biotransformation is actually a really challenging subjective of the untargeted metabolomics because, well, mainly it's really hard to detect the biotransformation product, especially if you're working on the in vivo system. For example, if you want to look into the how the drug biotransformed into the different types of metabolite, well, if you want to do it in vivo, you're gonna just you're gonna feed the drug to the mouse, and then you're gonna get the blood from the animals, and then you're gonna analyze them with the mass spectrometry. But then you're gonna get a tons of MS features there, but most of them are gonna be actually just metabolized from the mouse's own metabolic system. And really a small amount of MS features we're gonna be the metabolite from the drug. And of course, the amount of such kind of metabolite are quite smaller than the original metabolite they might have. And yeah, every time that happens, like whenever like you're working on like the microorganism culture or a microbial like community, they're gonna make up a tons of the metabolite, and then by transformation product, we're gonna be just a really small portion of the entire metabolome you can get from the mass spectrometry data. So yeah, drug metabolism study has always been a really important part of the drug discovery and development pipeline, but it requires a tons of the time and effort on the looking into the data and finding out some metabolite there. And I just wonder, like, how can we identify the metabolite from complex mixture, easier than the traditional approaches? And then, fortunately, during my postdoc, I could get some ideas about the important tools I can apply for such kind of the untargeted metabolomics projects. So yeah, as I introduced, well, I did my postdoc in the UC San Diego under the guidance of Peter Durstein. And he made up a really famous tool named the MS spectral similar networking or molecular networking. And mainly it just clustered the similar spectra based on the modified constant similarity and then like gather the similar spectra here and the similar cluster the similar spectra here and here. And it means that this similar spectra may be maybe they come from the similar structures. Of course, sometimes it's not, but yeah, usually the similar structure actually make up the similar the tandem spectra here. So, and it means that even in the case like you have tons of thousands of MS features, if you just compare the spectral similarity between these two features, you're gonna get a similar structures in here. It means that if you know what types of drug you want to track down in your data set, for example, you want to just find out the metabolite of the drug A, and then if you know the spectrum of the drug A, then it's the metabolite we're going to just gather in here, while the other metabolite we're going to just make up the other clusters in the spectral network as shown in here. And then also my friend the Ming Wang in the UC Riverside also made up a tool named the MassQL. Well, the paper we're going to be publishing in the Nature Method a month later, I expect. And then it's another really useful tool for finding out a specific metabolite of a molecule we want to track down. It's a universal language tool for finding out the MS data patterns, and then you can just find out what types, which spectra fulfill the condition you enter into the tool. For example, you can see here, like, for example, specific phase 2 metabolite case. So just entering this query, you can find out any types of MSMS scans having neutral loss of MOZ 162 in your entire data set. And if you're, if you're specialized in the natural product or like a tandem mass spectrometry, you're gonna recognize that it actually can be used for finding out any types of molecule have a 1-glucose the substructure in their own, the entire structure. So I'm going to introduce my representative study on this theme. So this was the study on the phenotyping of the fungal biotransformation of phytochemicals. And then this project started from my my curiosity on the nature. So one day I just walked through the university campus and I just saw some like some decayed the autumn leaves in the land, and it's the wonder like, okay, because I was like, I was trained as a plant natural chemist. I already knew that many plants produce the compound for defend themselves against the phytopathogenic fungus. So it means that plants always produce and accumulate a lot of antifungal compounds in their own body. And in the autumn, well, many leaves just fall down from the tree, and then some plants even just die before the winter come. So they just become just the dead plant body here. And then we already know that the fungi is actually the major composer of the dead plant body. And then we're gonna think about like, okay, then plants already accumulated a lot of antifungal compounds in their own body. And even when they become the dead body here, the compounds will not gonna just vanish to somewhere. Still is in the dead plant body. Means that the wood-decaying fungi should somehow detoxify the antifungal compound, which is already accumulated in the dead plant body here. So my hypothesis here was that the wood-decaying fungi were going to have some very specific system to detoxify the various antifungal phytochemicals, and then it was going to maybe an enzyme with the substrate promiscuity, because it's really similar to our liver enzyme, because we take a lot of toxic compound through our food or any types of the environmental factors, and then our liver can detoxify them a lot. If you know about the DMPK of the drugs, you're gonna know that only single cytochrome P450, the enzyme isoform in our liver can metabolize more than hundreds of different chemicals, because their substrate specificity is quite low. So I just hypothesized that, well, the wood-decaying fungi, we're going to have a little bit similar types of system in their own body. So we tested the hypothesis through a really well controlled experiment. So we got the 22 different wood-decaying fungus species from my colleagues in the Seoul University, and then we cultured them, and it's the 13 different phytochemicals, and it's the cultured for 5 days. And then we analyzed them with the LSMS, and then analyzed the data using the different tools I previously mentioned. For example, the molecular networking helped us to find out the really similar structural analogs from our data set, consisting of 322 lolita files, while the MSQL was also used for finding out some specific already known phase 2 reactions, such as like a glycosylation or other types of conjugation reactions. And then we could summarize the phenotype we could observe in the culture-based untargeted metabolomics run. And you can see that most of the 22 wood-decaying fungi could somehow biotransform the phytochemicals we actually fed to them. And then, well, but we could also see a lot of species specific reactions here. And then, well, we could just make up some patterns for it. So you could see the all of the 12 phytochemicals we used in this the culture-based screening, and then we could see what types of reaction the fungal species actually could catalyze on the compounds in here. And then we could find out the Lantus bromelis was the species which catalyzed the most of the various reactions on the phytochemicals as shown in this box. And the next page is about the detailed the procedure on how we could get the information about the phenotype. I showed you in the table shown in the previous page, because it was like a designed experiment. We actually know which data came from which species and what types of pair of species and the compounds in here. So we already know what compounds was just fed into this species. And then we look into the data, and then we could just annotate the multiple compounds, which is expected to be a metabolites of these compounds. So we could just compare the mass spectra in here. And we could say, okay, for example, okay, this compound was fed with the compound 4, and then we could see a spectrum which have like a precursor compound, which of which M of Z value is exactly the same with the compound 2, but the precursor have like a 132 Dalton shifts, and then we can expect that, okay, this compound is the pentosylated version of compound 4. And then we just look into okay, which species also have this spectra in their data set, and then we can check that, okay, these species can just catalyze the pentosylation reaction into the compound 4. So we just did this types of interpretation to all of the 322 data set, but yeah, it was not very time consuming because we already had like a really well interpreted molecular network, and then just it was like a transferring to the network itself to the table of the species in the compound pair as shown in the previous slide. So based on it, we could get So based on it, we could get a lot of hypothesis on the biotransformation reaction happening in the pairs of the phytochemicals and these species, and then we highlighted and then we focused on the one single reaction mediated by lanthanum bromelis, and which was the pentosylation reaction. Well, the reason was simple because, any kind of enzyme catalyzing pentosylation reaction to any kind of a compound was quite rarely known. So we wanted to focus on it, and we actually wanted to find out the enzyme which catalyzed the reaction, and then wanted to find out if the enzyme truly have a really broad substrate, the window. So at first time, we need to which types of pentosylation actually happening here. So we just scaled up the culture, and then so we culture the Lentus rulers in a more larger scale, and just fitted up the compounds there, and then just perform the chromatography based purification of a product, which on the compounds we already found from the untargeted metabolic screening, and then we could illustrate their structure using the NMR. Because I was actually trained as a really classical natural product chemist, so yeah, I know how to do it. So yeah, chromatography and NMR was done, and just we found out the pentose which was attached to the phytochemicals was actually xylose. And we could see that even in the larger scale, the Lentus bromelis truly attached the xylose into the flavonoid compound named the bicaline, and then also attached the xylose to the new lignan compounds named the magnolol and holokiol as shown in here. And then also we found out that sometimes it's just the xylose compound can be attached to the compound that is shown in here. Yeah, and then we also tracked down the time-dependent phenotype of the bicaline for the culture of Lentus bromelis, and then we could see that, okay, even it was quite interesting that even when we fed up the bicaline, which is a flavonoid, but a glycoside, normally it is found from a plant named the Scutellata bacalensis, and then in the nature, actually, the compound is a form of a compound 1 initially here, because the compound 2 is actually the toxic guy, but it's gonna be toxic even against the producer itself. So yeah, well, it's still like a hypothesis. Is this like a hypothetical in the plant science? But normally the botanicists thinks like, okay, plants just save their own toxic compound with the glycosylated form in their cell. And when they need it, they just detach the sugar from its compound and they make it to the toxic version and just release it to its competitor. So the compound 1 is actually intact form in the plant cell, and then we also found that the fungus actually detach the glucuronic acid from the compound 1, and then it was gonna become more toxic from the compound 2 in here. And then yeah, it's really well known that the microorganisms use the deglucuronidase, the deglucuronidation enzyme for like a carbon source scavenging. So they might the lentils formulas might detach the glucuronic acid from here to use it for their like a carbon source in their metabolism, but the product is toxic, so they against it attach the xylosate into the detoxification. And then we could actually see it in the time-dependent data, like the compound 1, it become the compound 2, and then those later compound just increasing when the day, the time goes. And then, so we confirmed that the fungus itself can attach the xylose. So we wanted to find out the enzyme which has catalyzed it. So we look into the draft genome of the Udiking fungus we used in our the metabolomics-based screening. And then we could find out that the 8 different UDP glycosyl transferase actually is present in the whole genome of Blanton's promolis. And as we could not expect which enzyme is actually the one that catalyzed the xylosylation, we observed it in the metabolomics experiments. So we also ran the RNA sequencing. And then fortunately, we could see that just only one gene is highly expressed in our cases. So we could find out the UGT3 was the candidate gene in here. And we performed the heterologous expression of the gene, and then we could confirm that this enzyme is actually the one which catalyzed the xylosylation reaction against the flavonoid we used in our untargeted metabolic screening. And also we could find out that, okay, this enzyme is actually have the substrate promiscuity, because it also catalyzes the xylosylation against the different types of compounds, including the neolignin and the other types of, for example, like isoflavone or like a catechin. Even it could attach the xylose into the synthetic phenolic compound as shown here, for example, like estimate or levothyroxine. These compounds are non-natural, but even the enzyme itself cannot attach the xylose moiety to these types of enzyme in our in vitro the assay shown here. So we finally catalyze this enzymatic character, and then we asked there is the nomenclature committee for this the types of enzyme called, and then the enzyme got a new name of the UGT66 because it was a founding member of a new enzyme family, and it was not very similar to any kind of the previously characterized enzyme as shown here in the phylogenetic tree. Only one enzyme was a little bit similar, but yeah, it was also xyloside transferase, but it was not like a enzyme with the enzyme with the substrate promiscuity. It was a part of a biosynthetic pathway of different types of microbial specialized metabolites that is shown here, and then amino acid similarity was quite low, even if it was lower than the 30%. So we could confirm that this enzyme is quite new to us, as shown here. And well, we did not plan this, but yeah, reviewer asked us to get the AlphaFold based the structure, the 3D structure of enzyme. So we asked, I just asked my friend Martin Steiner in the Seoul University to help me, and then he did a really nice job. So he calculated the 3D structure of the enzyme we found in here, as shown in here, and also, he just tried his new tool named the FOLSIG, which enabled us to search of a similar enzyme, but not based on the amino acid sequence, but search based on the 3D structure. I mean, the folded structure similarity based search against the protein DB. And then we ran it and then found out there are multiple similar enzymes to this enzyme. And then most of the similar enzyme actually are present in the wood-decaying fungi, but different types of species in here. But even the most similar enzyme was found from the genome of Traumatis pubescens, but their amino acid sequence identity was lower than the 60%. So we can not sure if these enzymes in the Traumatis were gonna catalyze the same reaction or not. So yeah, currently my group is working on to find out the enzyme homologue of this enzyme we found in this study, and then just to see if there were gonna be also different types of the detoxification enzyme of the wood-decaying fungi or not. So yeah, after confirming the character of the enzyme, we just went back to our initial hypothesis on the detoxification, like does the xylosylation actually reduce the toxicity of a plant's compounds or not? So we just tested the compounds antifungal activity, and yeah, if we could knock out this gene in the lentils bromolysis, it would provided a really nice direct evidence for our hypothesis, but normally engineering on such kind of a non-model fungus is really challenging, and we could not make up the knockout of the lentils bromolysis here. So instead, we just tested the toxicity of compounds against the Aspergillus flavus, as shown here, and then we could see that while the alkyl compounds exhibit the severe toxicity, while the xylosylated compound had a less toxicity than the original alkyl compound, and so as the same similar pattern was also shown about the neolignin compounds. And we also confirmed that these types of biotransformation really happen in the culture or like the colonization of this wood-decaying fungus on the exact dead plant body we used in here. So we just culture the lentil bromolysis on the dried material of a plant which have a compound 11 and 20, and then 12, and then we can see that the xylosylated compound 11 and 12 was actually occur in the metabolome of this the fungus on the dried plant material. So in this study, we could suggest that the untargeted metabolome is a really useful tool for tracking down the phenotype of the biological biotransformation against the xenobiotics, and even based on that data, we can just go back to the genome of a microorganism we used in this study, and just to find out like a previously uncharacterized enzyme as a demonstrating in this study. And we actually could recognize that this method can be applied to any types of xenobiotic transformation because, well, the method itself is quite global. So I worked with my collaborator who's working in the drug metabolism. The drug metabolism mainly by the human liver. So I worked with the Professor here New in the Hanyang University, and we also tested if the molecular networking based method is quite useful or not for tracking down the drug metabolite metabolism in the human liver. So we just perform a benchmark study, use a really well-known drug, sildenafil, and then we could we applied our data analysis pipeline into the in vitro data set of the for biotransformation. And we could find the most of the previously known metabolite in the data set. And we also could find a bunch of, I think like more than 20 of previously unknown metabolite of sildenafil in our data set. And the another challenging part of the drug metabolism study is like, okay, we can just find out like a M of Z value and the tandem mass spectra of a previously unknown metabolite. But it's really hard to get the exact structure of them. So for this, my friend Louis Felix helped me to do this. So we ran the rule-based candidate metabolite generation using using a multiple different types of softwares, and then we just put the generated structural library of a possible drug metabolite of sildenafil and just to match the expected structure against the experimental spectra we got from our metabolics run in here. And so we could annotate multiple previously unknown metabolites shown here. And then thanks to the in silico tools we use in here, we even could annotate like which fragments ion shown in the tandem mass spectra we're gonna correlate with the substructure of drug metabolite structure as shown here, which is strengthen the annotation through our in silico pipeline here. So I know that these types of data analysis pipeline we're gonna a little bit tricky, and maybe we're gonna be a little bit challenging for researchers who's working in the wet lab, because normally I know like how many wet scientists we're gonna feel about the bioinformatics tools in the mass spectrum, like metabolomics, especially when the tools are in like a code base, and then it's really annoying. And then okay, I cannot understand how we can use it, like especially in like through the Python or the R. So hopefully, yeah, we're preparing this manuscript, and then now our the protocol papers in the second round revision in the Nature protocol. So we're gonna be like a point by point guideline about how you can use the GMPS or other different types of compatible tools for annotating detection and annotation of a metabolite in your data set. So yeah, I'm not sure like how we're gonna go, but yeah, I just hope like this protocol paper we're gonna be published soon, and more researchers in the metabolomics field we're gonna use it and then take advantage of it. And yeah, just a few slides left, and then I'm gonna talk about the project now is ongoing in my lab. So this method, I think like this method we're gonna be more powerful when it's integrated with the reverse metabolomics. So reverse metabolomics is the concept suggested by the Peter Dressing like a few 2 years ago, and I think yeah these two papers became quite like a famous. And then the major idea is like in the normal metabolomics pipeline, we're gonna just run like get the data, and then we're gonna compare a group, for example, like a PCA or PLSD, or like any kind of a multivariate analysis, or like a volcano plot, anything. You're gonna find out some like the biomarkers in here, and you're gonna annotate the biomarkers to these structures. But sometimes it's really challenging because you're not gonna have any idea about the even when you look into the spectra, sometimes you're not gonna, you cannot find out the proper structure for them. And the reverse metabolomics is quite like different concepts. Like for example, if you can expect any kind of possible structure in the metabolome. For example, in these structures, Peter actually worked on the microbial, the bile acid conjugate. In his lab, how to say, like chemically synthesized the hundreds of different bile acid conjugate using the one-pass synthesis method, and then you're gonna get the mass spectra of standard you have. And then you can search it against the publicly available data set in the Metabolites or Metabolomics workbench or the Massive. And then you can find out that, okay, if the you made up the compound A in here, and then if you search the spectrum of A against the public data, and then if you can find that, okay, these types of serum data set actually have a spectrum of A, then maybe it means that, okay, the metabolite A we're gonna be in the human blood. So this concept is called the reverse metabolomics, and taking this concept, but a little bit modified version is ongoing in my lab. So recently I got a major grant fund for this project. So I'm gonna use this, but in the culture-based way. So my collaborator at Professor Dong Lee in the Yonsei University recently generated a human gut bacteria library of 44,000 strains covering the 237 species via culturomics method, and it actually covers the 20% of all the known species of the human gut microbiota. So we're going to culture these bacteria, and similarly to my previous study on the Kang fungi, we're going to culture them and then feed some compounds in here. Well, compounds can be like the natural product found in the food, like a nutrient or the drugs or like a pesticide or any kind of compounds. We're gonna feed them, and then we're gonna culture them for days, and then we're gonna get the mass spec data. And using the exactly same data pipeline, we're gonna know that, okay, this species can actually convert this metabolite into these types of structures. So we're gonna get the what types of reactions can be catalyzed by each the human gut bacterial species, and then we're gonna use this the information and then go back to the whole genome of the bacterial whole genome, and then we're gonna find a loss of enzyme which catalyzing the biotransformation reaction here. So we just hope that we can fulfill the two objectives. The first one is we're gonna use this method and we're gonna integrate it with the reverse metabolomics. So we're gonna get a loss of spectra of the metabolites made by the human gut. So we're gonna get lots of insight about how they're gonna be observed into the body, and then what types of tissue they're gonna be distributed in through the reverse metabolomics way. And another point is we're gonna actually find out a lot of enzyme which catalyze the biotransformation in the gut bacteria. I think it's really important because most of metagenome-based gut 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 bacteria study, most of the metagenome study actually gives annotation to the gene, but actually we do not have any idea about the specific enzymatic characters of such kind of annotated gene. For example, here, okay, this is like a hydroxylase, but what are gonna be a substrate of this enzyme? We have no idea about it. I think we need to get a lot of fundamental data and knowledge base on the like a substrate and the other types of enzymatic character of enzymes shown in the gut bacteria, and then it will gonna be helpful for further metagenome-based the gut microbiome study in the future. So as a proof of concept, we worked on it. We worked with the skin bacteria because it's like an easier version of the gut bacteria. So we cultured some skin bacteria and fed the phytochemicals. We targeted some phytochemicals which is often present in the cosmetics because it actually happens if you just put some cosmetics into the skin, then the skin bacteria are gonna somehow biotransform the compounds there into the different types of the chemicals. So we expected it, and then we could find that some skin bacteria actually change their structures, like a hydrogenation happens in here as shown here. Well, liquid charcot was actually underwent the hexosylation reaction as shown in here, and then it was quite interesting because any kind of the hexacylated version of a liquid-charcon A have not been found in the nature before. So we thought like, okay, if we can find these compounds in the any types of metabolomics data, we're gonna maybe a product of, from the skin bacteria, and then so we just search the publicly available data, and then we could find the loss of the matched MSMS spectra from skin swab data from 36 participants' skin. So yeah, we highly we think like, okay, truly the skin bacteria can change the liquid-charcon A to the hexacylated version, and as the liquid-charcon is a constituent of licorice, and the licorice is in many times it actually is used for the skin product. So maybe skin bacteria, turn them into the hexacylated version. So we could find out from the skin swab based metabolomics data as shown here. So anyway, yeah, this is the last page of my talk. I hope that I did not use the time too much. So anyway, so in the natural products domain, Untargeted metabolomics have been a very useful tool for like a sample and a compound prioritization for further discovery work. Actually, but I think like rather than the discovery, Untargeted metabolomics can play a really important role in the different types of studies in related to the natural products. So I think it's really a powerful method for hypothesis generation. For example, in cases I introduced to you, actually the metabolomics in the early stage, it gives a lot of insight about the phenotype of microbiome the biotransformation, which is like a starting point of finding out the enzyme related to the phenotype we observed through the untargeted metabolomics. And I think like Huong, we're gonna be introduce you another very interesting case that the untargeted metabolomics can actually give you an insight about how the metabolite is relevant to the microbiome interaction, especially in her case, in interaction between the fungus and the bacteria. And yeah, my lab is also working on a very different types of project related to natural products, and then untargeted metabolomics frequently plays a really important role in such kind of a project, in regardless of themes. So yeah, I always thanks to my lab members like postdocs and PhD students shown here, and I always thank to my collaborators and listed here, and thank you for invitation to the EMN of Natural Metabolomics Society.
Thanks a lot, Professor Kang, for your presentation. I mean, we are honored to receive you today. It was a very nice talk, combining showing how to combine metabolomics and different other omics with RNA-seq and until protein structure. I think it shows well how this combination can really lead to a global understanding of ecological processes. So it was very interesting. And so please do not hesitate to ask questions in the Q&A session. We will ask these questions at the end of the second part of this webinar. So yeah, do not hesitate. And now I will let the word to my colleague Diana Pinto to introduce our second speaker of the day.
Hi, greetings everyone. Thanks, Thomas. I have now the pleasure to introduce Huong Pham, who is a PhD student in Dr. Kyo Bin's lab. She joined the group in 2019 as the first graduate student of the group, and I am very pleased to introduce her. And please, Huong, the floor is yours.
Thank you for your very nice introduction. Can you see my screen? Today we'll talk about like cytophore, additional function of cytophore can relate the population dynamic of fungal bacteria community like by inhibiting the bacilli metabolites. So Cyto4 is microbiome's specialized metabolite, have functioned strong chelating iron, and to overcome the iron scarcity environment, the microorganism will release the Cyto4 to reuptake the iron. So essentially, Cyto4 was success with several multifunction, like protecting the producer from toxic, or acting as a signaling molecule regulating the production of the virus factor. So Cyto4 also can drive the interaction. So in the bacteria interaction, Cyto4 can work as a protector of the B subunit from frontal reaction, or Cyto4 also can draw the driving antagonist in direction in the Pseudomonas community. So additionally, the secretion of Cyto4 in a gut microbiome community show a growth driven ability of in the bacteria, but also a growth inhibitor ability in the pathogen. And in the bacteria and plant interaction, soil bacteria can produce Cyto4 and release the methane toxification, and then enhance the growth of the plant, including nutrient uptake, iron accretion, or homeostasis. But until now, not yet have any research talk about the function of Cyto4 between the bacteria and fungi interaction. So in our research, we 86 Bielefeld will co-control with the B subunits and use metabolomic analysis. We found that one of the Cyto4 named Bacillobacter induced by Bacillus subtilis, just induced in only co-culture, but not monoculture. So we just have a question go up so what kind of function of the Cyto4 in here? So our first expectation for this research is we can gain a lot of the antimicrobial metabolites because we have a lot of inhibition zone over here. But contrary to our expectation, a lot of specialized metabolite significant degree. And then we really quite confused. And then we examine the metabolome of the area where the confront is on the co-culture plate, confronting zone and Bielefeld chloride. And we observe the significant degree in the intensity of detected MS feature. So comparing it from 86 confronting zone with the data from arsenic in a pure culture of the 86 Bielefeld chain, we found out more than 7,000 feature out of the 9,000 MS feature have the ion intensity less than half of those in the arsenic culture. And the Bielefeld chloride also have a similar phenotype. And it is really quite interesting. And this trend could be seen in chain by chain comparison. And Bielefeld brasilium is one of the chain showing the most drastic change in the metabolome. So you will see the base big ion chromatograph here. Between the arsenic and co-culture, a lot of the metabolite significant degree, and we annotate them they a no compound, and we also isolate and use enema to confirm the structure of the velocolazen and 13-oxovelocolazen. The other chain, the other Bielefeld chain also have similar phenotype or co-culture with the B subunits. For instance, Bielefeld bicartum or SB99. However, some of the Bielefeld have the opposite, not 100% global phenotype. Some of the chain also show they increase the new peak, and the new peak already annotated as the antibacterial activity compound. To figure out which metabolite went up or down when the B subunit, we use the IAM 145 B subunit. So to convert the co-culture, so we use the mass spectrometry data, and then we run feature-based molecular networking, and we also use a computer-based tool to identify unknown spectra to do network annotation product operation with natural product atlas database. And this prediction have us pinpoint 13 cluster that represent a smaller molecular family some of the noteworthy metabolite we identify, you can see over here, is glycophenolate or imidazole alkaloid or tetracethyl methyltabenoate. They have very good activity degree here. And we also see that we confirm detected the bacillobactin, the free form, not bind with the iron in 47 co-culture, and we couldn't see the bacillobactin in a mono culture of B subunit IMA 1145 in the same condition. So based on that, we expect maybe bacillobactin have ability or inhibit the specialized metabolite of bellicinium in co-culture. So to confirm this, we choose an other strain. That strain number is 168. That the domestic strain, and it cannot produce a bacillobactin, and based on the inactive of the SBF protein. And we grow B subunit 168 with the bellicinium and use the same method which we did use in the brief strain. And we found out that, like, contrary to the co-culture with the B subunit IAM 1145, the number of increased MS feature in the co-culture with the strain 168 is more higher than degree of feature. So it's like quite opposite with the last previous co-culture. And for being sure, we choose the B subunit 168 knock out the DSPF. That gene, like, for being sure, like, they cannot produce bacillobactin in case. And then we try to grow a co-culture between bellicinium with the B subunit IAM 1145 and which can produce bacillobactin, and the other, like, B subunit, cannot produce bacillobactin. And we found out, like, velocozan and 13-also velocozan level significant degree when we co-culture with B subunit IAM 1145, but then they, like, have the similar level with the monoculture when we grow with the B subunit 168 or 8 mutant. And we also see the same phenotype were observed in the other metabolites in the other bellicinium co-culture with the B subunit IAM compared to 168 wild type or mutant. Like for, like, to investigate whether the interfungal specialized metabolism of the is attributable to change a transcriptome level, we perform RNA sequence analysis for bellicinium brasilienum co-culture with either bellicinium B subunit IAM 1145 and 168 knockout of the DSPF. So, as you see in the figure over here, among the coenzyme, we can see the enzyme FTMA to FTMH. They have, like, they encode for the velocozan benzene pathway, and they significant degree in the co-culture. So we can confirm that, like, the degree of production of velocozan and it allowed in the present of the bacillobactin producing by B subunit IAM 1145 is due to reduced expression of biosynthesis gene related to velocozan production in bellicinium. And because the velocozan benzene pathway, they need iron to produce product, right? But here we have a cytopore, so we think we hypothesize that maybe because cytopore by the iron that may make the specialized metabolism degree. That means, like, bacillobactin maybe inhibit the fungal specialized metabolism by causing iron starvation. So for confirm our, like, hypothesis, first we need to see the iron scattering phenotype by the Cas assays. With the pink color over here, that means we have less iron, and in the blue, that means we have the iron here. So when we co-culture with the B subunit IAM 1145, at which we have the bacillobactin, the iron scattering phenotype happen. But when we grow with the 168 mutant, we just normal. We still have iron over here, and the bellicinium will pretty well. And to confirm more, like antagonistic effect of bacillobactin again to the specialized mechanism, we culture Bielefeld in medium dilute iron to compare the level, the metabolite in the case normal media. So here we choose two conditions. In the first condition, we use the gel-acting agent. That means we can buy iron in medium. And then the second condition we can use like we make define iron defense, and then we also make define iron defense and plug iron in here. And the result is very interesting, like velucogen and 13 also velucogen, they significant degree in the like dilute iron environment, or in the like define iron defense, they like degree compared to like define iron defensive plus iron or the pediatric control media. So, okay, maybe we can think like, oh, iron is have really big role over here. We also try to test with the other Bielefeld, and we saw the same phenotype happen with the other strain. For instance, in Bielefeld vicatum, the structurally cyclo aspect A, or the averyantin of the Bielefeld SP99, they significant degree when we delete the iron in the medium. So based on that, we conclude like the metabolic inhibitor activity of bacillobactin were mediated by the iron starvation or abilisum coloni. But no side effect cheating, you know, like the heterologous utilization of side effect produced by different species, like the microorganism can use the side effect without produce them. Is like well-known phenomenon among the microbial interaction. So we just think like maybe Bielefeld have ability to use the iron bacillobactin complex or not. That is why we put bacillobactin in the media and then grow Bielefeld in a monoculture and co-culture with B-subunit 168 mutant. And really interesting, the velucogen amount of velucogen and the 13 also velucogen degree when we put bacillobactin in the environment, and compared to the deficient iron, defined iron deficiency, they quite similar. And we just think like not only bacillobactin, we also have the other kind of the cytophore, and we choose two more cytophore to work with this, like desferrosuamin B and the ferric brom, and we try to grow with the same method with the bacillobactin. And velucogen and 13 over also velucogen level also reduce order in the order cytophore supplement medium. So that means like Bielefeld Brazilium was enabled of the iron cytophore complex. So this finding support that the metabolic inhibitor activity of bacillobactin were mediated by the iron starvation around the Bielefeld colony. And one more question went up when you work with this research, like if the cytophore inhibit metabolites, especially the antifungal metabolites, so maybe it can have or maybe have a function or an population dynamic within a microbiome community, like try to coexist together or maybe protect the other microorganism. So we build up for unnatural community. So in there we can grow with the B subunits, like different strain like EMN 1145 can produce bacillobactin, or 168 or 168 mutant cannot produce bacillobactin with Bielefeld, and the third partner here is E. coli or Pseudomonas butida. And after like when you grow, there's a timeline for growing. After 7 days growing, if you see clearly, when we grow together with B subunit IAM 1145, like have the bacillobactin in here, the E. coli grow really well, grow together with Bielefeld and B subunit. However, they already covered or maybe die when we co-culture with B subunit 168 knock out the HBF. They just die or maybe just only Bielefeld. we also work with first E. coli and now it's Pseudomonas. And the similar result it happened. Pseudomonas grow really well. Grow together with the Bellicinium and B succulent IAM 1145. However, it's already died when we grow with co-culture with the B succulent 168 or 168 knockout. We also try to do the community with another Bellicinium, Bellicinium vicatum or BB99, and we also see the similar phenotype. The E. coli grow really well together with Bellicinium and B succulent IAM 1145, but it died in the other case. And it's also same happened with the Pseudomonas. Like under Bacillobactin, like cytopore up presents, the bacteria, the third partner, E. coli or Pseudomonas brutida, survive together with the Bellicinium and the Bacillus. So like in the conclusion with my research, like iron is really important for the biolysis process. They can help the Bellicinium produce antifungal metabolite, but when they connect with the B subcellulars, like the cytopore, the B subcellular induced cytopore, they cannot uptake the iron more, and the metabolite decrease. And this also is the first time, I think, the first time the cytopore will describe the external function, like they can like inhibit the toxic metabolite. And we also make the typical co-culture like unnatural community. So we try to demonstrate like the present or absence of iron deletion by the cytopore and decide which species would be dominant in the environment. So at the end, I want to say thank for my PI, like lab mate, and then the professor and work with us is quite a long time. So it's already finished. Thank you for listening.
Thanks a lot for this very nice presentation, this very nice example of how we can use metabolomics in this kind of field. It was really interesting for me at least, and I hope for all participants. So thanks everyone for the questions in the Q&A. I saw that Professor Kang was very fast to answer them. So thank you for that. I think we can just have some rhetorical questions just to get some participants to ask other questions for the remaining minutes. And indeed, I will have me some questions. Like the first one would be, when you mention your last experiment, how do you, like each enzyme have its own optimum with pH, with temperature, etc. So how do you manage to input those data when you have like 1,000 of enzymes and hundreds of xenobiotics?
You mean like how we can optimize the assay condition for enzyme?
Yeah.
So if you mean about our work on the microbial enzyme, well, so at the first stage, we actually the screening itself will not gonna be in vitro because we're gonna just culture the microorganism and just feed the xenobiotics into them. So mainly the reaction are gonna be inside of the bacterial cell, so we do not have to optimize the condition there. But well, if you want to confirm that the specific enzyme actually catalyze the enzymatic reaction in here or not, then obviously we need to purify them or just to get the the heterologous express them and purify them. Anyway, we have to run the in vitro assay for confirming the enzymatic character there, and then there is no like a true answer because you have no idea about the enzymatic character there. So because they were gonna be new enzymes, so you have to test multiple different conditions here, and then because it's actually the way to characterize the full character of a previously unknown enzyme, because we have to get the optimized parameter and have to report them to the scientific community. So we have to just test multiple different conditions.
Thanks. Sounds like a lot of
Yeah, because it's
work.
like yeah. Oh yeah, because it's the enzymatic characterization itself is the major objective. So yeah.
Yeah,
Maybe we can get, maybe we can be aided like by some computational tools. For example, now there are multiple types of tools which can predict such kind of parameter. Yeah, there are many teams like trying to predict like the enzymatic character, like the pH or different types of the things, or even like predicting the substrate using the machine learning. Actually, I'm a little bit skeptical about it, but yeah, maybe we can get some help for such kind of tools when we have to settle down the initial condition for a
yeah,
test.
definitely. And it's also represent a massive input for the community once you get this data, I guess. And yeah, I think another question I had, like, what is happening with catechin? I think it's what's the only compound that was not metabolized in your talk.
Which one?
Catechin, I think it was. Like there were one compound that
Yeah. Actually, yeah, it was not metabolized, and I think no idea. So basically, if you mean my slide in there were like 6 large X letters in there. So it means that yeah, the ion intensity themselves did not decrease. While the others were just a blank. Means that somehow they just decreased in the intensity, but we could not find out the proper the metabolized signal in our data set. So basically, yeah, the large X means that yeah, you can estimate that none of the microorganism cannot metabolize any types of xenobiotics. It means that they have no enzymatic reactivity against the substrate. While the blank means that, yeah, somehow they're going to metabolize it to something, but you know that mass spectrometry cannot observe all the chemicals. Mainly, you can only see like a chemical which can be ionizing to the positive or negative ion, while some compounds cannot be seen in both mode, especially in the ESI mass spec system. So we cannot track down such kind of metabolite. It's actually a really important point in the mass spectrometry based metabolomics that you cannot see every molecule in your data set.
Yeah, no, for sure. Especially for your next project. Like, what ionization mode are you using? The negative or positive one?
You always run both.
Okay. Yeah, well, usual. Okay, thanks a lot for your question. I think I have a last question for the second part of the presentation as well, because you also show the benefit of using NMR, combining NMR with mass spec, and that is really interesting. But I know that many members of the community kind of struggle of using NMR because it kind of people think that it takes a lot of time, and I think it does. But what was your like your feeling about it? How much time did you took, and what it bring to your project?
NMR, you mean?
Yeah. Because I think it's really valuable and really important for this project.
It's quite challenging. So basically, I'm a lab member, and so as we are the classical natural product chemist, I was trained as a classical natural product chemist. So I really did a lot of like a structural elucidation using the NMR, but I know that it's really, well, how do I say? It's really hard to learn at the first time. So I'm sure. So even in most of the natural product lab, well, NMR, sometimes we just get like we learn about it, but normally, okay. So what? So yeah, we're gonna feel like it, and the only way to get familiar with it is like you need to practice. So yeah, it's really tricky, but I really love NMR, and then I even sometimes in my lab also use it for metabolomics itself. Well, obviously it have a lot of limitation, like the low sensitivity, and it's a little bit hard to deal with the data. But but on the other hand, it have really, you know, the strength, like it we're gonna enable you to do the absolute quant for every metabolite you can see there. It's really important point of it, you know, it's really the large strength of the animal. So yeah.
Definitely. Okay, so thanks a lot again for your questions. I think for your responses. I think we do not have additional questions in the Q&A session. So I think it's time to end up this webinar. We were again very, very pleased to welcome you today. Thanks for your participation. It was a very interesting, inspiring talk from both of you. And thanks all participants. If you missed one part of this webinar, do not worry because it was recorded and it will be placed online as soon as possible. And as I said, do not hesitate to contact the EMN or to follow us on the social networks. And thank you all. Have a nice day. Bye-bye.
Thank you,
Nice
Dr. Pin
Thank you.
and
Thank you,
Huong.
Ying, for invitation.
Thank you.
Bye-bye.
Bye-bye.
Bye.
Bye-bye.
Thanks.
Have a nice day.